Welcome, everyone. I have a very special guest with me today, Dr. Sandell Sfanos, who is an associate professor of pathology, oncology, and urology at Johns Hopkins University School of Medicine. She is an amazing scientist who is studying the influence of the human microbiome on prostate diseases, including prostate infections and chronic inflammation in the prostate. And I am very delighted to welcome her here to talk a little bit about some state-of-the-art research that she has in prostate carcinogenesis. Dr. Sfanos, take it away.
Well, thank you so much, Dr. Ackerman. And I really appreciate the opportunity to tell you a little bit about some recent work that we've been doing that is exploring the effect of infections and inflammation on prostate carcinogenesis, and of course involves the human microbiome. So in my laboratory, we come at the microbiome and its relationship to prostate cancer from two different fronts. And I usually start off my talks this way by saying that what I'll be talking about today is direct interactions of the microbiome with the prostate and in prostate carcinogenesis. And here we're really talking about the urinary microbiome, and that is a thing, and I'm sure you've heard about it, but we're talking about the microbes that reside in the urinary tract, and that includes both the urethra and also the bladder. And here we're talking about microbes in the urinary microbiome that are getting into the prostate when they shouldn't be there. So this is infections that are occurring in a pathogenic type of state. We also study indirect interactions of the gut microbiome in prostate cancer, and I won't be talking about that today, but it is worth mentioning that your gut microbiome could profoundly influence prostate cancer development or progression via mechanisms such as drug metabolism, metabolism of dietary components, influence on cancer treatment and therapy, etc. So I'll begin my talk just to introduce the topic of proliferative inflammatory atrophy, because this comes into play a little bit later on. So this is, you might or might not have heard of, an inflammatory or an inflammation-related lesion that occurs quite frequently in the adult prostate. And this is prostate atrophy, but it's not atrophy in the sense of these cells are withering and dying away that you might think of when you hear the word atrophy, but it histologically looks like atrophy, but these cells are actually highly proliferative, i.e. the name proliferative inflammatory atrophy. So this is the stain for Ki67. These cells are actively dividing. They are responding to the inflammation. They are changing their gene expression patterns from being normal luminal prostate epithelial cells to expressing very pro-inflammatory cytokines. And for many years, this has been proposed as a risk factor lesion for prostate cancer development. And this idea has been championed by Angelo DiMarzo and colleagues that the cell of origin for prostate cancer might actually come from these proliferative inflammatory atrophy lesions. So the premise of this study that I'm going to tell you about today actually came from two seemingly unrelated science articles that came back in 2005 and 2006. One of which is very familiar to the prostate cancer field, which was the article that first described the fusion gene that occurs between TMPRSS2 and its transcription factors, namely ERG, which we'll be talking about today. And this fusion gene occurs in about 50% of all prostate cancers, depending on the population that you are looking at. The other science paper that came out described a genotoxin that is produced by E. coli and other closely related species. And this genotoxin causes DNA double-strand breaks in eukaryotic cells. So what is colibactin? So colibactin is what they call a non-ribosomal peptide synthase polyketide synthase. We refer to it as PKS. And there's the mechanism of action of colibactin is still being worked out, but currently it is being described as a cyclopropane warhead that breaks DNA strands. And in the context of cancer development, what was really important here is that exposure to E. coli or closely related species that produce colibactin can cause DNA double-strand breaks, i.e. ones that could potentially cause fusion events that occur in prostate cancer. And so colibactin was first described in uropathogenic strains of E. coli, but has later gained traction now in the colon cancer field because there's strains of E. coli and other species that contain colibactin that reside in the GI tract. However, this is a study here that was published in 2011 by Eric Oswald's group that looked at strains of E. coli isolates that had caused acute prostatitis in young men. And what they found when they characterized these isolates, that over 70% of the isolates are actually strains that carry this colibactin gene. So this is one of the studies that initially got us excited about looking at colibactin producing E. coli in the prostate in relation to prostate infections, because it puts it right there at the source where it could potentially contribute to prostate carcinogenesis. So our hypothesis was that bacterial infections, perhaps via colibactin, could cause oncogenic temperature gene fusions. And in addition to that, we hypothesized that these oncogenic gene fusions might occur in these early risk factor lesions that I told you about, such as PIA. So the way that we went about studying this recently is we identified a series of cases that had active bacterial infections in the prostate at the time of radical prostatectomy. Now, this is a really rare occurrence. This doesn't happen very often, and we screened several years worth of cases and developed a cohort of 15 cases that were very highly inflamed and suspicious for an active bacterial infection. And I should mention that all of these cases had pre-existing cancers that might or might not have been related to an infection many years prior. But the point of our study was to actually look at the non-cancerous areas of the prostate that were very highly inflamed and that had bacteria in them, so that we could look in human tissues to try to understand what happens to the human prostate during a bacterial infection. And what I'm showing you here is a series of techniques that we developed to detect bacteria in this series of 15 cases, showing you in situ hybridization for bacterial 16S ribosomal RNA. This is an assay for LPS, lipopolysaccharide on gram-negative bacteria, and LTA on gram-positive bacteria. But the moral of the story is these cases were very highly inflamed, and we confirmed that a subset of the cases had active bacterial infections because we could indeed see the bacteria in the tissues. So the next thing that we did was we went to look for the presence of TMPRSS-ERG gene fusions in the benign areas of these prostates. And we did this using an IHC assay for ERG, which is a readout that is used for TMPRSS-ERG gene fusions. And what we found surprised us. What we found was a very high frequency of ERG expression in PIA lesions in these infected cases. So ERG-positive PIA hasn't been described in the literature before. So the presence of these ERG-expressing PIA lesions, we think, was specific to the fact that these cases had active bacterial infections and all that inflammation occurring. So we found ERG-positive PIA and also low-grade PIN in about 50% of these infected cases. And this was a much higher frequency than we found when we looked at a series of 64 unselected cases that were not infected. So the frequency there was only around 5%. This is just a picture of what ERG positive PIA looks like. This is ERG IHC over here on the left in a PIA lesion. And then we also confirmed this with an in situ hybridization assay for ERG mRNA. So you can see the brown dots here is ERG mRNA expression in these lesions. This is a low-grade PIN lesion showing ERG IHC, positive IHC, and ERG positive in situ hybridization. And this is another example of an atrophy lesion where you see ERG positive IHC, and the brown staining is ERG positive in situ hybridization. And then the negative areas by IHC are also negative by in situ hybridization. So the next thing that we did is to further confirm the presence of TMPRSS-ERG gene fusions in these lesions was to use a tricolor FISH assay to demonstrate genomic rearrangements at the TMPRSS2 and ERG loci. So one of the most common alterations that you can see by FISH is a TMPRSS-ERG fusion via deletion of the interstitial region between TMPRSS2 and ERG. And the readout for that is loss of a green signal in this particular tricolor FISH assay. So wild type is red, green, blue, If you have a deletion, you get loss of one green signal. So long story short, we did confirm the presence of temperous ERG genomic alterations in these PIA lesions. This is just one example here. This is a PIA lesion. And hopefully you can see that in these cells, you can see the one red, green, blue signal, and then just the red, blue signal in the other allele, showing the loss of the interstitial region between TMPRSS2 and ERG. And all the arrows here show cells where you can visualize that TMPRSS-ERG fusion. Very importantly, we also described in our study that these ERG-positive PIA lesions are often directly adjacent to very early budding carcinomas. So if you look at this example here, this large region circled in blue is all ERG-positive PIA. And the little region in red is early budding microadenocarcinoma from these ERG-positive areas. Again, suggesting that perhaps the fusion event occurred in the PIA lesion and that this lesion is progressing to an early cancer. We also fully mapped these cases in 3D to demonstrate that the ERG-positive PIA lesions were not simply some sort of retrograde spread of an ERG-positive cancer. And in this particular case, you can see there are several different ERG-positive PIA lesions throughout this prostatectomy specimen. There's a little cancer down here that is ERG-positive, but it's a completely different fusion pattern than all of these other PIA lesions that we described. So this is not just retrograde spread of cancer. These are ERG-positive PIA lesions that are happening de novo in other areas of the prostate. And we found 3 cases with ERG-positive PIA where there was no positive ERG cancer in the entire case. So finally, we did a little bit of work getting back to colibactin to show that in 2 of the cases that we included in this study had regions of bacterial presence that were large enough that we could actually macro dissect out the tissue and then look to see if the infecting strain of bacteria was producing colibactin. And long story short, in both cases, yes, the infecting strain was a colibactin producer. This is showing colibactin production by in situ hybridization and also by qPCR. And then we also showed in a series of in vitro experiments using LNCaP cells that if you expose prostate cancer cells to, this is a lab strain of E. coli that either contains a back vector that produces this PKS colibactin or has an empty back vector, that if you expose LNCaP cells to colibactin, you get DNA damage, as you see here in comet assay with the comet formation when you expose them to colibactin. Radiation is a positive control. And then we also visualize DNA damage via increased expression of gamma H2AX in cells exposed to colibactin in this in vitro system. And then finally, we also show that you get DNA breakage at the TMPRSS2 and ERG loci using our FISH assay when these LNCaP cells are exposed to the colibactin genotoxin. So you can see breakage again between the TMPRSS loci and then also between the interstitial region and the ERG loci. And it's quantitatively shown over here that you get an increase in breakage at these loci with exposure to colibactin in prostate cells in culture. So in conclusion, we have demonstrated that there's a high frequency of TMPRSS2 ERG fusion events that we observed in PIA and low-grade PIN lesions in cases harboring active bacterial infections. And at least in 2 of the cases, we showed that these are infections that were from colibactin-producing strains of bacteria. We observed in several of the cases that these precursor lesions were in direct transition with early microadenocarcinoma. So we think that what we're visualizing in these tissues is very early cancer formation in the setting of a bacterial infection. Certainly our readout for cancer initiation in this particular study was via Tempers-2 but it is certainly possible that infections and inflammation in the prostate could contribute to other oncogenic events and in other mechanisms toward cancer development. But the moral of our story is that we propose that bacterial prostatitis should be considered as a legitimate risk factor for prostate carcinogenesis. And of course, this merits much future study, but just a few ideas here in terms of developing methodologies to detect prostate infections that might be potentially undiagnosed. And certainly, can we develop strategies to mitigate infections, specifically with colibactin producing strains of bacteria, as a prostate cancer prevention strategy. And with that, I'd like to acknowledge everybody that contributed to this study, especially Eva Strespa, who was a former graduate student in the lab who championed this current study that I talked about today, and of course, all of our funding sources that have made our research possible.
That was wonderful. Thank you so much for sharing this work with us. I had a couple of just quick questions that I wanted to ask, and you sort of alluded to some of this at the end of your talk. The isolates, the Escherichia coli isolates that you found in association with the cancers and from patients were all these colistin producing ones. Is there any evidence that an E. coli isolate that, say, did not produce that could be protective, or that there is no association in those circumstances, sort of the opposite sort of experiment?
I mean, I think that this is all, it's a great question and all to be determined. It's obviously it's hard to
find the negatives.
Well, find the negatives and to use human beings as an animal model. Right. They don't perform well as an animal model. So I think we need to be creative about how we're going to study this in vivo systems. It's very hard to induce prostate cancer in a mouse via an exogenous infection. We've tried this in the lab. Other reports and other studies have tried this as well. But it could just be that mice are not little humans, and that the mechanism of prostate carcinogenesis in a mouse is quite different. So I think we need to be creative about how we're going to study whether it's specifically colibactin producing strains that are pro-carcinogenic, or whether it could be any type of infection, and maybe even not strictly bacterial, but viral infections or fungal infections or parasitic infections in the prostate that certainly can produce a tremendous amount of inflammation, even though the infecting strain themselves might not be producing a carcinogen. Mm-hmm. But inflammation in its own right might be pro-carcinogenic. So I think it's a great question, but there's a lot of work to be done to really answer it.
Yeah, I mean, at least it would be interesting to know, or I mean, to continue this work, obviously, but I'd be interested to find out, if targeting in patients who kind of have these infections, where there is this mechanism of potential carcinogenesis going on. And obviously this is hard to study, but whether using these pro DNA strand breakage factors could be a potential therapeutic target. And any thoughts on, obviously you have the strains that do and do not produce that. Has there been any exploration in your lab of sort of inhibitors of this pathway, and looking at how that influences the generation of that chimeric molecule?
Yeah, I mean, these are all studies, it's a great suggestion, and studies that we have, thought about and we have ongoing in the lab. No data to report quite yet. But I think you touch on a very important point, in that, the men that we studied in this study, the inflammation cases, the cases that had infections, clearly these were all undiagnosed prostate infections, or else I'm not sure they would have done the radical prostatectomy if they knew that the infection was there. So I think there, in terms of a cancer prevention strategy, is a lot of work that needs to be done in terms of perhaps strategies to diagnose inflammation and maybe even active infections in the prostate that are either not diagnosed or are the symptomatic of something that's being diagnosed as something else. And there I could envision perhaps maybe, urinary types of tests for inflammatory markers, et cetera. But then also keep in mind, I started out the talk by describing this as a urinary microbiome problem potentially. So we've also found and I didn't present the data today, but in patient or a series of samples that we had from men without prostate cancer, that if you look in the urine, you can detect colibactin in a subset of men. So this might be something that's carried by bacterial species that are present in the urinary tract of men that don't even know that they're there. They might or might not be causing infections, but they certainly would have the potential to cause significant damage in the prostate and maybe even also in the bladder. So I think diagnostic tests for the presence of these organisms in the urinary tract would be important.
Not just the organisms themselves, but actually some of these virulence factors, if you can kind of use them as a biomarker for sort
Exactly.
of more pathogenic inflammation that could predispose people to such, carcinogenesis. And then lastly, I just wanted to ask, is there, any thoughts for how, this work might influence the urologist seeing a patient with some prostate symptoms coming into clinic, and how we might want to adjust our thoughts about how to approach that patient?
Yeah, I mean, that's a very interesting question. And again, I just have to refer to maybe, if you're working that and I'm not a urologist, so I'm not, I don't know if I'm the best person to answer this question, but if you are working that patient up for a potential case of acute prostatitis, for example, it might be important not just to find out what strain of bacteria is causing it, if that can even be teased out. But again, like as you just mentioned, maybe a further evaluation of virulence factors associated, because maybe that's somebody, especially with recurrent prostatitis cases of a strain that produces a genotoxin. Maybe this is somebody that you would need to follow a little bit more closely in terms of prostate cancer risk. Maybe we're not there yet in cause and effect and confirming our data, but it might be something that is important down the road.
Yeah, I think the more we learn, the less we realize we know. But I think it really is becoming more and more clear in multiple sort of dimensions that the strain of bacteria and its individual virulence factors, and this also goes for other microbes as well, plays a really critical part. And we just don't know enough yet to figure out how to apply this in clinical practice. That was an amazing talk. Thank you so much for being here with us today. I look forward to the next chapter in your research, and we'll follow it with bated breath. And once again, it's a pleasure to have you here today. Thank you again.
Thank you so much. I really appreciate the opportunity.